The advantage was really that we were able to build up a consortium in Europe, and we were able to deliver an infrastructure for brain research that is EBRAINS. So what came out of it? And you find these brochures with more information on the website. There were many results which were quite impactful. For example, measuring consciousness. Here the colleagues have developed a method that allowed to measure the activity of the human brain, and based on a theory, they were able to quantify this level of consciousness. So this is Tononi, who is probably the most famous person. But this method went into clinical practice. For example, in Leuven, there's a colleague, Stephen Loris, who is working with patients in coma. And for these patients, it's of course extremely important to understand what do they perceive. Yeah, they do not have any reactions. Yeah, and but sometimes there is some remaining consciousness, and they recognize, for example, close relatives. And this level of consciousness, therefore, has an important meaning of how to treat these patients, as you can imagine. There are robots, for example, with brain-inspired movement and condition. This is my colleague Rainer Göbel from Maastricht. He has developed a model and a simulation of a dexterous hand that has quite a number of joints. So in that respect, it is very human-like. And the way how this hand is being controlled is based on imaging experiments that told him something about the circuits, about visual motor coordination. So he directly implemented it into this hand motor control, and he can show that the hand then learns faster and converges faster than traditional methods. And now he is attempting to teach the hand to play piano. Can you imagine? So it's really cool stuff, and it's inspired by the brain. And powerful supercomputing for the community at large. So that is also very important. And sometimes I get the arguments, why do you need this? I need my laptop, maybe a little cluster in the institute. I would say that modern neuroscience is not, you cannot think about without large computers, because the complexity of the brain is, of course, so huge that you cannot put it into one little computer. Yeah, you can simplify, you can model, and you can also have simple models. That's fine. But if you want to simulate and to monitor the whole pipeline, this simply doesn't work. So let me give you one example of what the HPP did. So we had different simulation engines. This is how we call it. So there were simulation engines modulating and simulating neurons and microcircuits. And one of the simulation engines, this is Arbor, has been developed here on site together with our Swiss colleagues. So Arbor is modeling single neurons with very high levels of detail. And then you can simulate, for example, small networks of neurons, and can simulate their electrical activity. So you have very detailed microcircuit models. And then, you simply have the problem that there are many neurons, 86 billion, and you cannot model everything with such a detail. You also do not have the information. So the way you do it, you simplify the neuron models, and for example, use the nest simulation in order to model and simulate point neurons, but more, and many of them. Then at the brain level, you can have so-called neural mass models, or you can also have connectome edges. So if you conceptualize the brain just as a network with nodes and so to say edges between them, then you have also a rather simplified model. So the payoff is that you lose the accuracy and detailedness of the neurons, but therefore you have the entire brain. And when you bring them together, you can inform, for example, the higher scale models by information from the lower scale models. And this is something what we started in the Human Brain Project. And then the last is really your modeling behavior and have, for example, these sensory and cognitive models that help to develop neurobotics. So when the HPP started, we had only single modeling and simulation approaches, and I can tell you that the colleagues did not friendly look to each other. Yeah, so the NEST people said, well, this is the way we have to approach it. If we have too much information, then we lose the entire view of the human brain. And so to say, the systems level people argued, well, we need to understand behavior, and a cell model is too detailed. Yeah, we do not need it. So there are all kinds of discussions, was friendly discussions always. But the idea is not to have one model and try to, so to say, to beat everything, but take advantage of different models and then connect them in a good way. There is, of course, another aspect when we think about learning and plasticity and adaptation, then we have to look to the cerebral cortex with the different layers, and there have been different models proposed, how feed forward and feedback and how horizontal connections are arranged. But Peter Rolfsema, a colleague from Amsterdam, he also connected this physiological activity and anatomy with neurotransmitter receptors, for example, with acetylcholine. And what we did here in the institute was to measure the concentration of receptors of neurotransmitter sites in the cerebral cortex. And then, so to say, you can again connect both, and Nicola Palomero is going in more detail about the receptors of the human brain. And this kind of research ended up in a clinical trial that is now ongoing in France. It's not yet finished. So 13 epilepsy centers participated with 400 patients. And the idea was to make a prediction based on a model and a simulation of a particular patient, then feeds the model with information from the atlas, for example, from physiology of this patient, from neuroimaging of this patient. So putting together the theory, the physiological and imaging data, plus atlas anatomical data, creates, so to say, a more personalized model, and that was used in order to predict where the surgeon had to remove pieces of the tissue in epilepsy patients. And this is always tricky because you want to remove enough so that the patient is seizure-free. Certainly you want to remove the seizure, but you do not want to remove too much because you do not want to remove healthy tissue, obviously. And to find here a good compromise is extremely difficult, and we hope now that Victor Josa's model developed together with other colleagues, of course, that this helps. So there is a brochure of Spotlight, what came out, because sometimes it's questioned what really came out. I would say there are scientific breakthroughs like the consciousness thing, yeah, or the epilepsy. And certainly we were pioneering brain research, have built a collaborative network, and have also initiated new projects and collaborations. So this is a little bit what we ended with, and at the end we developed a roadmap for digital brain research, and we identified 8 different fields where we would see in the next 10 years there is a lot of dynamics, and we need to contribute to this roadmap. And you see it goes from the atlases, modelings and simulations, neuromorphics, and many of the points that are raised here you will see. But the interesting thing is, I mean, we had more than 100 authors, and they came 50% from within the project and 50% from outside. So it's not just our personal opinion, I would say, so it's really a broader a broader thing. And when the HPP finished, we were lucky to receive a new grant, which was much smaller, and which was had the target to further develop this research infrastructure to advance neuroscience and brain research. So it is an infrastructure grant. It's not so much a research grant, but of course, any infrastructure development has to be driven by neuroscientific questions. So we try to combine both. And this project started in 24 and it finishes now in 26. And I would like to invite you to go to the website where you find all the tools and services and the data and also examples to work with. So what does it mean, EBRAINS? It's a digital platform, I would say. It's a distributed platform. So some pieces are being maintained from here, from Munich. Some pieces are maintained from Berlin or from Paris or from Oslo. So it's really a European platform. And we enable research at different levels. You know now why this is important. But we have focuses on certain services and tools and use, of course, very much computing. And this type of, so to say, infrastructure, you can focus on 6 different elements. So this is the atlases of the human brain, monkey brain, rat, and mouse. Then there is a big chunk of data and knowledge services where you have all kinds of data that you can identify, so they are fair, and then use for different, so to say, for different other purposes. Of course, all the data that are in the atlas are listed here. Then we have the medical analytics. This is about medical data collected from a number of European hospitals. Then there's modeling and simulation. I have mentioned it already. There's a collaborative platform, so you can log in and then work together in a collaborative space. And then there is a computing infrastructure where you have access, for example, to supercomputers like the one we have here in Munich. But again, there are more supercomputers and other computers in Europe. There are neuromorphic systems, very interested. So you have all kinds of different computers available. The data, this sounds easier, honestly, than it is, because in order to describe data in a way that they are really accessible and findable and searchable and reproducible, that's a lot of work. And many neuroscientists, they do not like to annotate their data in a way so that you can do it, because it's a lot of work. Yeah? Annotation of data is a lot of work. Imagine you have all kinds of different experiments, and you should describe your data in a way that you can reproduce the experiment, at least understand it. So therefore, we have developed with a Swiss colleague a system of a knowledge graph. And I show you what the knowledge graph is. It's a database, but at the forefront, you have a user interface that tells you what kind of data we have. So there are more than 1,000 data. There are models, metadata models, software, web services, contributors, and sometimes the data are really complex data sets by themselves. So behind the data set, there is a database, if I may say so. It's not an Excel sheet, obviously. Yeah. Then you can search for this data. You can look what is freely accessible, what is under embargo. You can have a look, is this brain data or something else? Is this human data or monkey or rodent data? Is this in vivo data or postmortem data? Is this female or male data? Yeah, which kind of experiment, MRI or 2-photon imaging? So there are all different filters where you can search the data, and this is openly available. You can just go in and have a look. And this is a little bit how this database looks. And we decided not to go to so to say, simple database, because the relationships between the data are rather complex, and they can model it like this database, and all the links, they indicate what are the different connections between the data spaces, and the numbers indicate how much entries you have. So that's the way how the database looks in the background, and there's an open mind schema behind that also has been developed by Jülich and Switzerland and Brussels together to describe all this data. Even licenses, softwares, and so on. So there's really a lot of data, metadata behind. And I mean, brain data are organized topographically. Yeah, it's important whether the data comes from here or comes from there. So the atlases, they host a lot of data and help to put it into a topographical context. And Jan Bialy from Oslo in particular is developing an atlas tool for rodent atlases. So there's a so-called Waxholm space, which is a rat. There is a link to the Ellen Institute, who have a very nice mouse brain atlas. And then there are different parcellations of the rat brain atlas, plus there are very nice software tools that help you to work with the atlas. So if you're interested in understanding what a certain concentration of a certain cell type is in a certain region, what the numbers are, then you can use the Quint workflow. So the rat atlas is almost complete. For the human atlas, that's a bit more tricky because the human brain is bigger. And this bigger also means more complex, and it's more difficult to map the brain. It takes longer time. And this is, I mean, the difference is really if you have a Toyota Corolla or you go into a Ferrari in terms of efforts, yeah, not else, but it's simply different. You have to find different approaches, and in the human brain, you cannot, you cannot do many experiments. Yeah, human brains vary a lot, and this variations is captured in so-called cytoarchitectonic probability map. So here we have mapped 10 human brains with respect to these two areas in the frontal pole, and the color code indicates the probability which you can find when you register all the 10 brains in one reference space. And blue means that there is a high variability, low overlap between the 10 brains. And red means low variability and high overlap. So these probability maps, they show how variable brains are, and they show where you can find a certain area in the human brain. So you can imagine you do not have to map one brain, but you have to map 10 brains. That's a bit different in the rat, honestly. There is not so much variation. The mouse does not have so much variation. Practically, they are very similar. The human brain is very variable. And this is, so to say, influenced by the folding pattern, of course, but also the microstructure itself is very variable. So we are now working for 30 years on this atlas, because one area per year, 2 areas, 2 years per person. And we have now identified more than 270 areas, so you can multiply it. It's a lot of men or women years that goes into that. And that results in this type of atlas. And of course, we are happy that this now features in Grey's Anatomy. Not in the movie, in the book only. Still. So why this atlas is different and why it is new. It has the probabilistic maps that I have shown you. And for simplification, I have labeled here every area with a different color, but behind are the 10 brains always. So the information is behind. But then the atlas that we have developed also goes to the microstructural level, to the so-called big brain space, which is a 3D reconstruction of one human brain at 20 micrometer spatial resolution. And here you can see microscopical details, and later afternoon we show you how these brains will be cut. And you see the extent of different brain areas, and again there are some white spots because it's so time consuming and you cannot automatize it in the way we would like to have it. But having both brains in one and the same spatial context and in the same framework, you can really jump back and forward, go from higher resolution, higher accuracy, lower resolution, but probability, and links this to imaging. So that's a little bit the idea. It's not one brain. So to say, it's a little universe, if I may say so. So we are offering all these different cytoarchitectonic maps, and they can be analyzed using the ZIPA toolbox, and Timo Dickscheid is later on explaining what you can do with the data. But the idea is really to have something like Google Maps, but for the brain. So with Google Maps, you can identify where I am. Yeah, Jülich Research Center, and you get, for example, the coordinates. You get also the number of the street. You get also, so to say, an implementation into a broader context. You know, Jülich, North Rhine-Westphalia, Germany, middle of Europe. You can also get the climate information, if you like, raining very often. Yeah, you get the mean temperature. So Google Maps helps you not only to understand where you are and identify the pathway to Stadthotel or to Aachen, but it also helps you to work with the data, and you can then compare Jülich with Nice, for example, or with Palermo. Yeah, and you see the differences. So, but localization is, so to say, the basis which allows you to make this comparison. And this is enabled by Zebra. So Timo and his group, together with our friends from Padua, they have now developed a new approach to analyze data coming from patients. And this is functional and structural neuroimaging data. And the special thing here is that there are so-called 5G data. So there are 5 different modalities. So one patient with a neurodegenerative disease or a stroke, for example, is scanned 5 times, always the same person. And then you have, so to say, everything in one. So these poor people, because it's a long protocol, they have to be very quiet. But then you have everything, and you can exclude that methodical differences of your measuring method influence the result. Yeah, and since brains are so different, you never know, is this because of variability, is this because of your method, or is this other factors? So these 5G data are extremely important, and they are running based on Zebra. And the other, this is together with Rainer and Goebel, this is a desktop application that helps to compare the cytoarchitectonic maps with functional imaging data, but you can do it very soon on your desktop. And why this is important, I mean, it's not always possible that you log into the internet. Sometimes you are behind a firewall in a hospital, for example, and then you need to get the software and the data to your desktop behind the firewall. So that's a little bit the idea. And then we have a collaboration with FreeSurfer, and those of you who are interested in functional imaging know that FreeSurfer is a very cool software tool to extract surfaces of the brain and do all kinds of structure function analysis. And together with Bruce Fischel and Doug Grieve from Harvard, we are extracting from our postmortem brains the surfaces where we have mapped the areas. So this is how it looks. These are postmortem brains. We have extract the areas, we inflate the brain. Here are the contours. And the idea is, again, to predict with higher accuracy where a certain functional activation is in the brain based on these cytoarchitectonic maps. And we are really excited about this collaboration, although the political situation is a bit weird. Honestly, we wanted to have an NIH grant with them together, and then Trump said, no money to Europe. But we are still collaborating. But this is how policy sometimes influences science, of course. But here, I think we can move forward based on this collaboration. And I would also like to show you another example of impact of the cytoarchitectonic atlas. This is a work from a colleague, a neurologist who is now in Cologne. His name is Andreas Horn. And Andreas Horn is developing models of deep brain stimulation of patients. So when you have Parkinson patients and they cannot be treated by drugs anymore, then they get in electrodes deep into the brain, for example, into the subthalamus, subthalamic nucleus. And what he is modeling the trajectory of different electrodes based on the big brain. So this is our high resolution template in order to predict the best localization of electrodes stimulating the brain of Parkinson patients. And they have found, or they describe the so-called sweet spot. So it's really important and accuracy measures. So 2 millimeters, 2 millimeters away, and the effect is not as good. So for deep brain stimulation, it really plays a role where you are in the brain. Not only, but it really plays a role. Okay, this is now another application from a completely different field that's from colleagues in Heidelberg and Bern. And they are using modeling and simulation approaches in order to develop new neuronal networks with very high predictive power. And they are also developing photonic neuromorphic devices and just really nice publications for the sake of time. I do not have time enough. Hello, Timo, to explore more. But have a look. These are really cool papers. And another application that comes out is from Maurizio Corbetta, and he has studied brains of patients with glioblastoma. And what we know now that glioblastoma is not just a tumor who's invasive and occupies blood vessels, but glioblastoma cells, they use the neuronal network in order to distribute along nerve fibers. So very tricky methods. And he and his team have identified methods to predict based on diffusion matrix in these patients the time which these patients will survive. So glioblastoma is a very rapidly developing disease, and these patients have a life expectations of 1 year, 2 years. So it's really a bad disease. And some respond to certain drugs and some do not respond. So what a doctor can do is, so to say, to give the drug only to those who respond, obviously, because any drug has side effects. And this method helps him to understand who are the groups and how to stratify it. So the epilepsy trial is still not yet finished, but what we see is that it's extremely successful. And I will not go into detail. You will get my slides. So we hope that this year we get the results. And now the same method is being used for also other diseases like schizophrenia. And also the idea is to develop models of brains of schizophrenic patients and then to simulate when a certain drug is being administered and then simulate before the patient is getting the drug, how this patient will react. And that's another project that is, so to say, an outcome, I would say, also of the HPP, and it's now dealing with psychiatric disease. So how do we look now into the future? We update this roadmap and have collected, therefore, different contributions from researchers in all Europe. We have launched a roadmap process. And just yesterday was a submission deadline for proposals. So we have received 100 proposals from the community, just proposals. It's not about money. It's about formulating where do you see a need for neuroscience to go to, and then to develop the right infrastructure, develop the right funding mechanisms, and so on and so forth. So we will publish these contributions and then make a kind of summary and have a roadmap symposium in May in Munich, and will then, so to say, publish this roadmap proposal and share it with the community. And this is also very natural about science. Yeah? Science is changing very quickly. And now in particular, based on AI, I mean, you all observe it, how quickly the capacities of AI networks are being developing. I'm deeply convinced that, so to say, major breakthroughs in the future can be done when we better understand the brain. And it's not, so to say, this brain-inspired neurobotics or AI will play, hopefully, not hopefully, I'm rather convinced, will play a bigger and bigger role in the future. So this makes neuroscience also so attractive, also as a topic to do research and to be there. And at the end, I invite you to discover EBRAINS in order to make your research more efficient and more effective. And I'm a bit over, I would say, but I wanted to give you this introduction and prepare the next talks to you. And thank you for your attention. And of course, any questions, I'm available. Yeah, thanks.
Yes.
Yeah, please, Charles.
Thank you for this presentation. I think we're all here convinced that this is a huge work and it's going to be super useful for the community and for the future. I have one question though. Is there any ongoing debate regarding the bioethics and ethics about all this research?
Yes.
It's also a rising concern, I think.
Yes, of course. I mean, and not only now. So again, in the Human Brain Project, we had an independent sub-project for ethics, because you cannot start with ethics debates when everything is there. Yeah, sometimes you have to implement it from the very beginning into the tools, and the whole data pillar in EBRAINS has to be ethically informed. And one of the results is that we have certain access modes for sensitive data, which have other requirements than other data. So this is one example. But also, I mean, all the debates about consciousness that are now ongoing, general intelligence, this is an important debate. And as every ethics debate is always, we have two different sides. Yeah, you do not want to stop any AI application because you need it for medicine. There's no question. Yeah? You need to accelerate it. But how much do we need? How much do we want to interfere? Another example is deep brain stimulation. There are these closed loop stimulations where the stimulation goes based on the needs of the patient. That means you have an electrode, which is also a detector. And then in dependence on what is going on, the electrode is being adjusted. So that's, of course, very much interfering with the brain. So there are many questions, I would say, and we, in the EBRAINS project, we have advice from ethics, from an ethics committee, and we have these discussions with the field. There is an ethics debate now, I think, yeah, this week or next week. This RW Schneeweiß, it's now on the 19th, maybe. I can share the link. It's yeah. Hope I do not forget it. So please remind me in the afternoon.
I continue on this data. If I can continue on this on the sensitive data and stuff, does whole project like that have to create a more unified European regulation on ethics of sensitive data, or do you have like a lot of issues because every country has different?
Okay, there is a European ethics regulation which was very much pioneering the GDPR. Yeah. And the national do not go beyond, so below. So they are not. You have to apply to the European. You can do more in your countries if you feel, but you should not do less. So that's the idea of the GDPR. Indeed, it's not always, I mean, sometimes it is helpful, sometimes it's really a problem. Because there are clear restrictions, and share data is extremely difficult and also creates a lot of paperwork, which is not always good for science. So you want to compare data across hospitals. Very challenging, no question. So also here you have both sides of the medal. You have a good legal rule, I would say, it's fine, but sometimes it makes your life very hard.
Hi. I have a question about funding. I guess you have like European funding, but do you do like collaboration with private enterprises or something like that?
Not yet enough. We do have European funding. We have some funding from the institutions, like Research Center Jülich is also contributing, not money, but I mean, for example, FTEs or space and so on. Not yet enough, but we should go in this direction, and we are actively trying to collaborate now with partners from industry or from smaller business. So that's on our plan, but not yet really functioning, unfortunately. Netherlands and Belgium are better in that respect, I would say. They have much more power in this collaboration.
Because otherwise we don't have time for a quick break and then come back. So
thank you for the talk. I mean, at least from the outside, the project is some similarities to the Human Genome Project, right?
Yes.
This took also much longer, but now the cost really dropped and we can sequence one gene a day for really few months. When I speak to persons now doing some connectome research and so on, they always say, oh, it's so complicated. So when comparing these two projects, in which phase are we in the human brain project or the technological advances? So did it improve or get faster to scan the brain?
Well, the first comparison with genome is very attractive, but at the end you have the genome and it is there.
Yeah.
And then you find out that's more difficult because what you see on the genome level does not always go to the phenotype and to the behavior and to the disease. And only a few diseases are, so to say, monogenetic diseases. But still, the progress that we have seen after that in the genome project, when we identify at-risk people, when we identify now better and better drugs acting at the genetic level, shows that this was the right way. But it's 30 years. Yeah, and the Human Brain Project has started with a very broad basis because we also did not know what is, so to say, what is the right way. There is no single solution like genome sequencing. That's the problem. Yeah, you have physiological, you have anatomical, behavioral, cognitive, you have all kinds of different methods, not one. So we tried to build a platform that serves as much as possible with having a few foci. And the foci now are going towards, I mean, as I said, stroke and neurodegenerative diseases and some glioblastoma, I think. So schizophrenia and this other project. So we try to focus and make here the biggest impact, but the data that you need is much broader, of course, than only coming from this disease state. But I'm convinced that this is the right approach, and at the end, everything is freely available. You can take it and build on that, so it's not lost. Also, the project is, so to say, finished. We do everything in order to continue to provide the services, offer the data, and so on.
Do you also make a technological advancement, or is it still a lot of manual labor to continue to get the results? Because I think, right, as the Allen Institute, they have manpower, right, to do the atlases.
Yeah. And it's mouse. Yeah, it's mouse.
Yeah.
So it's not the Ferrari, honestly, it's mouse. Yeah, but we are trying now, of course, using AI in order to automatically label as much as we can, so that not the poor scientist has to develop. I mean, Maurizio Corbetta told me he was sitting 1 month in labeling some 1,000 or 1,500 data. But first you have to understand how to do it. Then you have to develop the tools, then you have to develop the quality control, and then hopefully it goes. But this is the future. You cannot do everything by hand.
Let's thank Professor Amos for the talk. It is my pleasure to introduce you to Professor Timo Dickscheid, who will talk to us about the atlases. Timo is also working here at the Forschung Center Jülich in the institute where Professor Amos is also located, and I think you have been working on these atlases for so many years now, Timo, from bringing the concept together to the structure and implementation. So we are
delighted today to hear more from you on how building these atlases actually looks like and how to use them for the applications. And then I leave the floor to you. Thank you so much.
Thank you, Sandra, and welcome everyone. I'm actually quite excited about this. Sandra just told me how the things were going and the kind of program that you are following. I think this is really, really cool. So it's actually something that we could, I think, EBRAINS will benefit a lot from people like you who have a more regular training and maybe focused interest to bring things together. I find that quite exciting, actually. So I'm happy to talk to you here. I'm not so happy that my screen is arranged in the wrong way, but this I will fix right away. And it is further complicated by my Zoom windows here. There we go. So this looks better. Now I have to move the mouse back to my screen. Here we go. Yeah. So yes, I'm Timo Dickscheid and I'm actually a computer scientist. So I'm not a neuroscientist, but I have spent now many years with the neuroscientists over there in the building and in Europe, so that I have kind of a neuroscience touch, but I'm a computer scientist. My actual background is computer vision and image analysis, and I started here in Munich to analyze microscopy images. And this is how I got step by step more and more to the atlases. In the Human Brain Project, then computer scientists were quite needed because we also had to build infrastructure. That's what we look at now. So that's how I somehow slipped in and enjoyed being at the intersection of computer science and neuroscience, so to say. I hope I can give you some, let you benefit a little bit from this now today. Yeah, I will talk about the brain atlases, and as I understand, you already have quite an idea of the what EBRAINS is, and Katrin Ammunds already showed you the bigger picture, but very certainly also some details about atlases as well, because she's of course one of our main persons behind the brain atlases. So there could be a bit of redundancy, since I will also of course pick up on atlases and EBRAINS, but I try at these stages to be rather brief. Yeah. If you have the feeling if something is going too fast or too slow, you can just raise your hand and fast forward or rewind by waving hands. I will be happy to adopt to this. All right. So at that stage, you probably know already what a brain atlas is, but it is a good idea to start with the very basic and obvious things, just to make sure that we're on the same track. So when we talk about atlases, we usually mean three things that make up an atlas, and that is a reference space, actually a coordinate system with an image that tells us where is the anatomy in the coordinate system, and that is similar to the Earth in a way, where we have the globe. Then we have maps that are defined in a reference space, similar to the Earth, of course. And then there is a taxonomy that tells us actually basically the names of the mapped regions, but usually also relationships between them. That's why the taxonomy is very often a hierarchical tree or something like this. So it tells us how we call these things and in a basic fashion how they are related. That's kind of the very basic ingredients of atlases. But our approach and also our ambition, EBRAINS, goes a bit beyond that. And the very obvious way in which it goes beyond is that, just like for a map of the Earth, we assume that there are different ways how you could map the brain, since it follows different principles of organization that are related, but that can be studied also separately. So we have different maps. And we also have additional aspects. If we start with this, so when we have an atlas, we usually want to know which brain regions are defined at which location. That's the very obvious fact. So we want to find somewhere, and then we basically here indicate by the color, we see what the region there would be. But then, especially in the human brain, there is a huge variability in these regions. So each region is quite different in different human subjects. I guess that many of you know about that. So we want an atlas also to encode this variability in the most reasonable way. As we will see, usually it is not easy to do that in a perfect way, but there are ways to capture the variability depending on what we do. And then usually these brain regions that we describe, they follow, they are mapped according to a certain principle of brain organization. And actually, we are also interested, what is this principle of brain organization that actually makes up the boundaries of that region? So what is it actually? And in this case here, they have been mapped by microstructure, so by the arrangement of cells, cytoarchitecture. I'm pretty sure that Katrin Amunds already told you about that. So actually, the information about these brain regions actually carries us to a higher resolution, because the information, what makes up a boundary in this atlas, is actually defined at the micrometer level. This is actually not what we see here, which is a standard reference brain volume, the MNI standard volume at a millimeter resolution. But the actual borders here, they have been defined at micrometer resolution. That's where actually the neuroanatomists have drawn the borders. So there is a cellular detail which is at a completely different scale, spatial scale, than the actual atlas, that provides us the descriptions of what makes up these regions. Yeah. In this case, that's not always the case. Sometimes we also have reference parcellations of the brain that have also been measured in vivo in an MRI at millimeter resolution. That's also possible, yeah. But in this case here, the actual information is at a higher level of detail. It's of course relevant if you want to interpret these brain regions. It is relevant to have access and to know about the cellular detail, because that's what makes up the atlas. So when we build an atlas system and we want it to be comprehensive, we have to make a link between these scales. It is actually not sufficient to just send someone a nifty volume with these maps here. Yeah, there's a lot more. And this you cannot send simply by email to someone, this stuff here. That's why we think an infrastructure is really important for atlases. And what we see here is now cytoarchitecture. That is, the Jülich Brain Atlas is based on cytoarchitecture, and here we go to a higher resolution in terms of the arrangement of cells. But of course, there's more than only cytoarchitecture. Besides cells, we have nerve fibers, we have neurotransmitter receptors, different ways of connectivity in the brain, functional aspects of the brain. So there are a lot of different modalities which also play a role to understand the brain, which should be linked. And what we want to do in this atlas is also to create the links with these different modalities by providing additional parcellations, but also by providing links to measurements in the brain that are regionally specific and that describe these different aspects of brain organization. That is our ambition, and that's how we went into developing the EBRAINS Atlas services, with this ambition. And I try to explain to you now how we did this and how we can work with it. Because in the end, once we have such a system, which is more or less a collection of data, a very broad one, the question is how you work with it. Yeah? And what we have built in EBRAINS are tools that you can actually look at it. Very obviously, you want to see it. Yeah? But also tools to program and script with it through Python. And of course, sometimes you want to run analysis or do more complex things, or maybe do complex things in an easy fashion. So you also want some tools to perform analysis with the atlases without programming stuff yourself. And on these three levels, we have developed solutions in EBRAINS, and you should find such solutions partly with different level of technical maturity. So that's basically the way out. Yeah, we have an ecosystem with atlas reference information and data that is linked to it. And now we have tools that you can work with it and access it. There's a whole second part of the story, which is why there's an empty space here. That is, how does the stuff actually come in? How does all this information come in? Because we cannot assume that at a certain point in time we have captured everything and we're done. But this is, of course, will always be incomplete. We can never capture enough information about the brain to model the variability, for example. Yeah, we will ever and ever be able, with new devices, to acquire data at higher resolutions, for example. So this is actually a living system, and we need also a way to make sure that scientists can contribute by bringing information into the system. And this is why in EBRAINS we also provide tools to anchor data to atlases. So if you have new data coming in, there are workflows that help you to actually integrate them with the atlas ecosystem. And I will, in my rather short presentation this morning, try to explain and show you quite a few of these things. Not in full depth, because there are also tutorials coming up for you in the afternoon, and I think also on other days. So I try to give an overview and give you some key ideas. What we need to do all of this, we need reference atlases that have a microanatomical precision to be able to do all this integration. Then we need many data resources that we link with this atlas, what we just saw on the screen. We need an infrastructure that can host all these contents, because we are here talking about many terabytes of information that need to be somewhere. Yeah? We need software tools, very obviously, to work with it. And actually, I'm not sure if we have time today for that, but what is a big part of my own work is actually, we also need data analytics and AI to turn all this information into actionable knowledge. So there is a lot of development going on in data analysis to create the atlases, but also to work with these atlases. Maybe we can have a few quick glances at such things. But the focus, of course, will be on these 3-4 points now in the next half hour. So this EduBrains atlas, I think that Katrin Amundsen has already explained it to you, but I want to give you a quick run through here again, because it is an important basis to understand other things that we are doing. So for this EduBrains atlas, is based on cytoarchitecture. We are cutting donor brains into ultra thin slices. In this case here, 20 micron resolution. So if you have a single brain, that's 7,000-8,000 such sections will be created in the lab. That's actually really happening over there. So you could almost look, no, if that building wouldn't be there, you could even look into the lab where they are doing it right now, probably. And then they are stained. We typically for this atlas use a Nissl staining, where the cell bodies become dark, and that's why you see very pronounced the cortical mantle, the cortex here appearing darker than what we call the white matter, because that's where the cell bodies are sitting. I'm not sure how much background you bring, whether this is trivial, but in the brain we have mostly here the white matter, the connections, so nerve fibers, and mostly here in the cortex the cell bodies. Yeah, and that's since we have stained the cell bodies to become dark. We see here very clearly that the cortex appears dark. And then this goes in a microscope, is imaged at the micrometer resolution. And what you could actually see is that between different brain areas, you see significant differences in how the arrangement of the cells is. Here it is very obvious. Even I, as a computer scientist, can easily see that. Katrin Amundsen can distinguish areas where I think it's all the same, but she can see it. But here we see it very clearly. This is the secondary and primary visual cortex. And you see that here's, for example, a layer with a bit higher density of cells that is not pronounced here. And also for example, the presence of larger cells here, the larger cell bodies here is more obvious than here. So there are a few properties that you can here in this case easily see that this is different. Yeah, very often it is more subtle, but it can be measured, and it has been done over decades by neuroanatomists. And if you do this in different brains, so it has been done then for these two areas, let's say 50 to 100 brain sections in 20 different brains, and then you can put these two-dimensional annotations of the boundaries back into a 3D image of the brain. And what you see there is actually how different these are. Yeah, so these are now 20 different postmodern donor brains, and this mapping has been done. And for the primary and secondary visual cortex, there is not much you can do wrong, because it's very clearly visible, and you see here very clearly how different it is in the different units. Yeah, it is always in the back, and it's always kind of that they are encapsulating each other, but the actual size and position, also relative to some of the foldings in the brain, is not so similar. It's quite different. Yeah? And that is why it's important that we have to capture this variance. And that's what's being done. So we do this mapping in typically 10 to 20 brains for each region. Here I've only shown 3. And then we register them into a common space. usually the MNI reference space, and we can compute then a probability distribution. Yeah, that tells us here where we see red, that basically means here in all the brains that we studied at this position, it was always the same area. But here where we see blue, it probably means that only 1 of 10 brains on average, the area actually extended so far. Yeah. So we see the variability. And some other regions are more crisp, sharper in the structure, and here it's quite smooth. There's a lot of variability. And what we can then do from all these, we have then, let's say, 300 of these probability maps. Then we can compute a so-called maximum probability map that tells us at each place what is the area with the highest probability here. So this is quite a huge simplification actually, but that's what you mostly see in the end. Yeah, that's what you mostly see and use. But for you as upcoming experts, it's actually good to know there's a lot behind. There's much more that you can use if you do a study. You can do much more clever things than just assigning these maximum probability voxels to your data. Let's say you have a functional activation in the brain and you want to see whether this is quite exactly one brain region or multiple. It would actually be much more wise to compare it with the probability maps than just with this. Yeah, so that's good to have in mind. And I have to speed up. And so that's the one thing. The other thing is we try to provide access to some of these postmortem brains at the full resolution, the single brains at the full resolution. And this is here the big brain where we did this in 2013. We built a 3D reconstruction of the complete brain from all the sections and tried to make this accessible. Hey, Nicola. So that means actually in the atlas, you have different representations. Yeah, we have a very high resolution single brain with areas mapped. We have these probability maps where we see the statistics of mapped areas, but then it's already summarized at a millimeter resolution. And we have also maps on the brain surface in the FS, in the free surface spaces. And everywhere we have the same areas defined. And of course, we can go between these different spaces because we know the corresponding regions. And typically, we also have spatial transformations that come from image registration algorithms that tell us what is the most plausible coordinate in this space that belongs to this space. So we have links between these different representations, single subject, high resolution, and so on. All right. That's an example of a complex atlas, of a complex map, yeah, that is defined across different spaces. But I told you, we want to link data to these atlases that actually characterize what the regions are. What is actually there? What is actually the cell density in a region? And this is the atlas itself doesn't tell us. It just tells us here are two regions. They have a different characteristics. But of course, we want to know what are these characteristics now. So we need more data that we link with the atlas, and that means that is a lot. I'm just showing some examples. Of course, we are interested in the actual histological slices that tell us, for example, something about the distribution of cells. We could have images that with color, this is an immunostaining that show us in different colors what are groups of different cell types, for example. We could have, you will see that this afternoon, we could have images that show us directions of nerve fibers, images that show us how neurotransmitter receptors are distributed across the brain, and Nicola will certainly tell much more about this. I guess she's the mega expert on this topic, sitting there already in the back. So these are just examples. There's a lot of data that actually explains. And from these images with this data, there is derivative data that actually summarizes. So for example, you could have data set that summarize at a certain place in the brain, what is actually now the density of the cells in a certain layer in the cortex, in a certain quantity. So then you have actually quantitative numbers. Then you have actually in the end a table of numbers that is relevant to a certain location in the brain or to a certain brain region. Yeah? And that's what you are in many cases interested in. Yeah? That's what you want to know. Here's a brain region, and this is the typical density of the cells, hopefully with a standard deviation. The receptor densities that Nicola will certainly also talk about, you also have the densities of certain neurotransmitter receptors. You have standard deviations, and you could have this in many, many different brain areas. And this was produced with the EBRAINS Atlas tools, this overview here. You could have connectivity matrices that describe, for a certain subject that was measured in vivo. What is the structural connectivity in terms of the number of streamlines connecting to brain regions? And then you can have many of them for many different subjects. This is all real data that you have in EBRAINS here that describe you how is a certain brain region connected with others. You could have gene expressions that were measured in a piece of tissue in the brain, such as you can get them from the Allen Institute. They also have a certain coordinate and tell you something about brain regions. So these are just examples. Question is, how do we link them with the brain atlases? And there are three fundamental ways how you can link data or content with the atlases, at least how we do it. And the very obvious way is the data are actually images, and then you use image registration algorithms to position them, to actually superimpose them with a reference frame. Yeah, so actually say this block of image measured in the image, it belongs here, so that's where it is. That's image registration. It's actually based then on images. You could also have data that was measured carefully by an expert in a piece of tissue, for example, and you get a table with some measurements, but the expert has carefully reported in which brain region that was measured. For example, here in the primary visual cortex, that's for example how we get the data from Nicola typically, many cases. So then we get a table of measurements, and we know from the study this is really representative for a certain brain region because it has been carefully localized. And then it's actually linked to the atlas by the name of that brain region, not by the coordinate. And there's a very obvious third way. You really have a coordinate. That is, for example, the case for these gene expressions from the Allen Atlas, where we know they took a very tiny piece of tissue in the brain and they have taken a note of where in the standard MNI space that has been. So these are three very fundamental ways how we can typically build a link from a piece of data to the atlas. Yeah? Name of the brain region, image registration, or coordinates. Now back to EBRAINS Atlas services. Katrin Amunds told you a lot about this. We have brain atlases as one category there. We have a big category on data and knowledge. That's where you can share data, search data, and so on. There are also medical analytics tools, modeling, simulation. You will hear about this from other people. There's a collaborative platform, and of course, there's a compute infrastructure. I'm talking about this and the links to this. So when we go to the EBRAINS page, ebrains.eu, which is actually currently has been modified quite a bit recently. So I looked this up this morning again. But you find very prominently here a link for brain atlases. I could even very briefly do this. So when you run ebrains.eu, you arrive here. Oh, you don't see that. You just have to stop the presentation for this. Now you see it, yes. You arrive here, and then you can say data tools and services, or even click here. Takes you to the same page, and you see your brain atlases, medical analytics, modeling, simulation, and so on. You've certainly seen that. We click on brain atlases, and you see we have different species here. I'm talking a lot about human, but there are atlases of monkey, rat and mouse brains as well. Even different monkeys now, yeah, macaque and marmoset monkeys. And here you find tools and software, and you find here also links to look at the atlas viewer that I will talk about. So there's all the information actually ready for you here. So as I said, we have atlas of human, macaque, rat, and mouse. There's software, how you can access it. There are tools to bring new data in and tools to analyze data. I'm jumping here a little bit. If you look only at the human atlas, you will also see there are different maps, not just the cytoarchitectonic maps that I talked about. There are maps of fiber bundles, for example, or functional organization. It's all there. And in the afternoon, you will get a tutorial where you can look at this in more detail. We have worked quite a bit in EBRAINS to establish the technical foundations to standardize the way that we express atlas. And this is a paper that has been, I think, last 2 years ago, probably, mostly driven by our collaborators in Norway, where we have tried to formalize what actually makes an atlas: reference data, coordinate system, a set of annotations, a terminology that matches roughly what I showed on the first slide. But here it's now quite technical and quite formally written down which things are actually which concepts do we actually need to describe an atlas. And such ontologies are really important for us because all the content that we have in an atlas, we will later on look here in the atlas view what's happening. But as I said, all the content of an atlas are actually data sets that are described with rich metadata. And if you want to describe metadata, you need some standards how you organize the metadata and how you structure it. So this is really important. Yeah. So when you deal with these atlases and EBRAINS, it's more than a rule of thumb. Actually, in most cases, what you see is a public data set with rich metadata, and many of them combine. So when we look at such a brain here, the template space is actually a data set, the maps are actually a data set, and these probability maps are also their own data set. So everything you see and you touch is actually a data set that you can also find independently of the atlas. Yeah, and you find all of that in EBRAINS. You find in the atlas services, you see everything organized in an anatomical way. And when you go to the EBRAINS data services, you find everything as separate data sets with much more information linked. Yeah, and this is connected. That's the whole idea. We're connecting data sets and content together to build such an integrated anatomical reference system as the atlas. And these data services, the core of these data services is what we call the EBRAINS knowledge graph. That's where we have all data sets, models, softwares with metadata and the links between them. And you can search and find it there. But if you use the atlases bring you there automatically as well. If you have your own data, there is the EBRAINS data services provide data curation teams. So you can really go there, request curation for your data, and then you will side by side work with a data curator to prepare your data and bring it in. It's a very clear defined process. Now let's have a quick look at these software tools. When you go to the online viewer, and we can probably just do that instead of looking at the slide. So I'm here back in, I just click here on Launch 3D Atlas Viewer. And if you're not fast, it will bring you to the human atlas first. But here at the bottom, you see you can select different species. You can select different reference templates, coordinate systems. You can select different parcellations. As we discussed earlier already, I can maybe increase the font size a little bit. Yeah. So here you see when I click here, I could select the monkey atlas, the rat atlas, mouse atlas, the marmoset atlas. I'm staying here, the human. If I click here, I can select two different MNI coordinate spaces, the free surface space, and the big brain model. That's the big microscopic brain that I showed you. I can select between them here. And here on the right side, I can select different parcellations. Here's one for cytoarchitecture. There are parcellations that show cell site, functional architecture, fiber architecture, and so on. So the whole idea is actually already implemented in this online viewer. Although the online viewer is quite a, it is quite a simplification since it has to be online accessible and easy to use. But you can actually here just click a brain region, and then you see this probability map that we told about. Once you selected the brain region, you find here information about that brain region. And very importantly, you find here a link with the DOI. When you click there, it brings you to the data set, which is just said, everything is a data set. Yeah. So there's a link, there's a DOI, you click on it, and here's the corresponding data set. In this case, for this probability map that we just selected. And then it says here features. So here you find data sets that are related to that brain region, data features. And you see there are some that have to do with the cellular architecture, molecular architecture, connectivity. Yeah. If I just, for example, take here a molecular one to make a nice link to Nicolas' work, here is this receptor density fingerprint that we saw on the slides. Yeah, the receptor density fingerprint for area V1. I click on it, and what you see here, here's a description of that data set. Here is a DOI. If I click on the DOI, it will show me the data set. But I can also look at it right away here, because the Atlas software knows how to load the data set. It can itself load the data for you, and we see here already the densities. Yeah, so these are standard deviations and mean values of densities of different neurotransmitter receptors measured in this brain region. And you can download them, but the important thing is it's just a data set. You see here the link. I could click on the link, and here we go. It's a huge data set. You see who built it, who's responsible for it. Here are publications, and everything is described here. The atlas makes it easy for you to see all of this in the anatomical context. If I dismiss this, just as one other example, connectivity. If I choose here connectivity, the brain regions will be colored by connection strength to the selected region. We selected the primary visual cortex, V1. So you see here areas that are colored by connection strength in terms of functional connectivity to the selected region. We could also select here structural connectivity, streamline counts, and you get different colors. That's a different data set. And again, the connections that we see here, that's a data set. So we have again here, here's an information sign, and I can click on it, and you will see this is the data set that has been published. Yeah.
The connection, all the images that you're showing, is it for the 10 brains that were analyzed, only for one person?
No, that is different per data set. So the regions that we see here, these are still the regions from the EduBrains cytoarchitecture atlas based on these 20 brains. You can see that here, because we are still looking at the EduBrain parcellation. That is cytoarchitecture parcellation. The connections between these regions, it's a different data set. Yeah? So the connections that we see here, that's why we have these information buttons. The connections that we have just selected, you see here, they are based on structural and resting state connectomes of a healthy cohort. So this is from neuroimaging. Yeah? But the computation of the connectivity has used the atlas to average the connectivity information. Yeah? But as I said, everything is a data set, and not all the data sets come from the same person and the same study. The commonality is that they all refer to the same atlas reference ecosystem. Yeah? But this is now a different data set. The connection strengths, they come from neuroimaging. I think in this case, even from the Human Connectome project cohort. But this you usually find, you see that here, yeah? It comes from the HCP cohort. Yeah? So it's really a lot of information that you're dealing with. You have, with a few clicks, we have now here touched already 5, 6 rather complex data sets. Yeah. But the important thing is you have all the links to the data sets and all the detailed descriptions there. You can always find it, look it up. Yeah.
But there is an option over there, the subject and the average.
Yes, yes. You can also look at the average and load the average, but this will take a bit because now it has to average them all. Yes, yeah. But this is rather customized. So there's a lot what you find here, right? And depending on what you have in your hands, you get your different context, right? Right. Oh, you asked me for the wrong thing. Yeah, the average is really taking a bit. Yeah, yeah. You can, when you look, you can see here it's even some information about tracing connectivity, but I think that this is actually refers to a different parcellation. Yeah. Okay. Since we are a little bit short on time already, just very briefly, I want to show you. So this was this showed you links of different data modalities with the reference atlases. We have not seen this traveling to the cellular detail. I very quickly want to show that. So if you would zoom in here now, and you can do this with a scroll wheel, or I do it here with my touchpad, you see that we are here in the MNI space. It has a resolution of 1 millimeter. So when we zoom in, when I press Q or when I click here, it hides the parcellation. And you see there's of course not a lot of what we can see, because that's an average reference brain. The millimeter resolution, we don't see much when we zoom in, yeah, of the anatomy. But if we now switch to the big brain, here, there's 2 things will happen. It changes the coordinate system. It chooses this big brain model, which lives in a different coordinate system. But since it knows in the background the spatial transformations between these, it will actually try to position the big brain at that same location. As well, you notice that we are here in the back of the brain, in the occipital part currently, quite zoomed and very much zoomed in. And it will preserve that. So now I click on the big brain. It tells me some information that it was not super exact and so on. cannot. But you see, we're here in the big brain now, approximately at the same place. Yeah, but now we are at this level of detail in big brain, we still see a lot because that's now a microscopic image data that we see here. So here's a lot of detail. We can here even zoom in further. Yeah, and here we see the cortical layers, a lot of stuff. This is a data set. Has a size of approximately this image approximately a terabyte that we are looking at now, and it just goes here through my Wi-Fi. I have no cable connection, so it's quite easily accessible. Yeah. And I can even here, with a hold shift, I could even do oblique cuts, so non-standard planes, can look at non-standard planes, which is quite interesting if you want to, at a certain position, go into a orthogonal direction relative to the surface and so on. And what we also see here, besides selecting brain regions, we could still search here in the space for data. So here we see now data sets that have been linked to the space, not to the brain region name. So you see here's obviously here's some lines that tells us there were images that have been anchored to this space. And I can select them here. Yeah, so here are some brain sections that I can load that are at the position where I was just looking at. And now I have here a brain section that has an even higher resolution. Yeah, really high resolution. Yeah.
Sorry, how did we get there? I lost it.
You will get a tutorial in the afternoon by my colleagues, but very briefly, I was just going to the big brain space. Maybe I was a bit quiet. So that's how it looks like. Yeah. I zoomed in, and what you then see here on the left is it says, oh, here are 5 spatial features. When this is closed, you will still see here a little 5. You click here, and then I just selected here's 1. Yeah. I clicked on it, and then it moved a little bit to move exactly there. Opened it here, and what I then did is, since this was a coronal image section, so it is in that plane, it is actually only here. Yeah. And what you could do is you can maximize this view. Here's a button. Also here, you see, everywhere you can say I only want to see that plane. So I clicked here to see only that plane. Yeah. And that just makes it. I could also have just closed this but of course, if you look at section, and here with space, you could actually cycle through the three views. Yeah. And then I have this very high resolution, and you can here really nicely see what you had on the slides, these cortical layers. You can see that everything nicely yourself. Yeah. You will hear a lot more in the tutorial this afternoon. I just wanted to give you a quick idea. Going back to my slides. So we have now seen the online viewer. The online viewer is, what is nice about it is very immediate. You don't have to install software. You don't need to learn a programming language, nothing. You just click around and it should hopefully will just work. It has several drawbacks though, since it can do so many different things. So it can actually, it provides access to very different kinds of data and everything. It is not as simple as some software that does only one thing for you. That's one you may consider the problem or not. Another thing is it is not nicely reproducible. So if you would actually use this for your research and you actually find some images there, of course, you don't want to do a scientific study where you take a screenshot and put that in the figure of your study, because no one else would take the screenshot at the same position. Yeah? So if it's what we did here is an explorative way of accessing the atlases, but it's not very reproducible. But you could make it more reproducible. One thing is you can actually store the URL of the viewer, because the URL encodes exactly what you see. So you could actually copy the URL, send it to your mates, and if they click the URL, they will see the same thing, yeah, up to screen resolution and so on. But of course, if you do it in a more serious way, you would want to do the same in programming. And that's what you could do. So there is, besides this online viewer, which we call software by Zebra Explorer, there is a Python client that can do very much the same things. And here's a little bit outdated screencast. So nowadays it would look a little bit different. But you see here how I am myself just writing a few lines of code that do the same stuff that we just saw in the online viewer, more or less. Yeah, I select the area V1 and the M&I space and the probability map. I look at the probability map. Here I have a receptor density that I queried based on this region that I selected. You will see this in detail in the afternoon. There will be a tutorial, so you don't have to write it down. Yeah. And here we go even to the big brain space and extract data at a much higher resolution. And this can all be scripted in a reproducible way. You will be able to access the very same data and everything by writing a small script. And this is nice if you want to use it for the paper also, because you could actually put the script there. You could say, if you use C++, here's a script how you get the data. Yeah. And you can still provide all the DOIs, which I showed you. You can still say the data sets I use, they have these and these DOIs. But if you want to see the exact same thing, here's a script of Python code that actually does it for you. And what I did here is actually a small piece of code that pulls and displays a cellular resolution brain section. Very much what we did in the viewer. But now you have a script that you can share. I will soon end. Just to show you, this is all documented. You will see this in the afternoon. But if you go, if you look here at the tools, I could tools and software in the Atlas on the web page. And if you search for Zebra, for example, then you find here Zebra Python. And if you say launch online, which is a bit strange, we are not launching Python online, it actually brings you to the documentation. And here in the documentation, you find all sorts of code examples. So you can just click on code examples. You actually find almost everything you saw. You find here multimodal data features. I stay with the receptors as an example. And here's a documented example of the code that you can download and run. It works. That actually shows you how you pull that. Yeah? There's many examples online, so it's very easy to find a piece of code that works and download it. Everything here has been executed before it goes into the documentation. All right, and as I said, there's a tutorial this afternoon where you learn more about this. So that was Atlas. I jump a little bit. I want to show you one example how you could use this Python. So with this Python software, you can do more than what we did in the online viewer. It is more powerful. And one typical workflow that I think is quite interesting for many use cases is you come with a certain location in the brain. Let's just assume you did an fMRI study. So you had a subject in the scanner and they were doing something, and you computed the activity map of the brain. So you saw at some place on the surface there was a strong activity going on, or maybe in two places. And now you want to know what's it. Yeah. Now you have a location. You have an image with a location. Maybe you click the peak. Yeah. And now you want to ask the Atlas what is there? What is the brain region? What makes it up, and so on. And this is actually nicely supported by the software. So a typical workflow would be you have a coordinate in a standard space, as you see here. And then Zebra could you do a probabilistic assignment to brain regions. So you get a table for your coordinate, which tells you this is with a very high probability, this is the primary visual cortex, but it could, with a lower probability, it could also be a different area. Yeah. So you get a table for this. And with these tables, you can load the maps, the corresponding maps, probability maps, and so on. You actually get a nifty files then in Python for this. And you could use the region names and the maps then to find data. So from there, you could actually run a query, and you could say, what is, if I assume it is in this brain area, which has the highest probability, what are the typical molecular cellular connectivity features in this area? Yeah, and this would in Zebra Python, this would just be 6-7 lines of code to do that, given such a coordinate. Sometimes instead of the coordinate, you want to bring the image. So let's assume you have this activation map from your functional imaging. So you really have an image, what is shown here. Well, then the software would also support you. The software would help you to actually split the image into components that are used there, and these components could also be used. So the images could also be used to do the exact same stuff. You could also assign the images to brain regions and do the query from the images. Yeah, and you could even use the images themselves to run these spatial queries. Was there any data in these coordinates independent of the brain regions? Yeah, and such a workflow, which is quite complex, you can do this very easily with the software, and it is described online. And what you can come up with if you do this for 2 brain regions, for example, what I just did for 1, you could collect all those features and put them side by side. And then you could actually see, oh, the connectivity of my 2 regions is actually quite different. Or the gene expressions of some candidate genes are quite different and so on. So this is such types of studies are very much simplified by the software. Okay. For one of these last cases, so if, as I said, sometimes if you have powerful software, which Zebra is, it also means that if you only want to do very, very simple things, sometimes you would love to have a software that does this one thing in a very simple way and cannot do other things, but this one thing is very simple. And for some very common operations, we also try to build tools that are very simple. And this is one that is just coming up now. It has just been developed by Rainer Goebel in Maastricht, where if you just have an fMRI scan and you just want at a certain location say what is actually there, the brain region, this is a desktop software that you could install on your computer that works in a very, very easy way. It cannot do more than that, but for this, it is of course very convenient. Yeah, and there are a couple of other solutions that you find in EBRAINS that use atlases to do a single thing in a quite simple fashion. But you find such solutions on the web page when you go to atlases and select tools. At this stage, I would stop, not to be too late. I started later, so please forgive me. But now I think we are still fine. I will stop here, and I still have still here for questions, depending on how quickly you want to start. Yeah.
Yeah. Thank you for the talk. Very, very, very interesting. So my first question is that are all the brains healthy brains, or do we have like Alzheimer's brains, like disease affected brains?
So the most important information is it depends on what you're looking at. As I said, we looked at many data sets, and this is actually a different story for each of the data sets. If you ask for the atlas that I've shown, these are postmodern brains of healthy people, but of course Nicola also again knows much more on this topic, but of course these people died. Yeah. So it is a question in how far are they. Well, what you know about these people is that there is no known disease, right? People looked at the brains, and there was a medical doctor who basically said there was no known disease like Alzheimer's or so. It was a healthy person that died maybe of a heart attack or something. And usually these brains are prepared, hopefully quickly after death, so that there's also not a huge degradation of things. But I'm not a neuroanatomist, so I have to be a little bit careful what I say here. For the atlas, it's healthy brains. And most of what we model in the atlas describes the healthy brain. Of course, sometimes when you find a data feature, it could be, since these are just data sets that have been linked, it could be that it's a data set of a diseased person, but refers to the same brain region. But then it would be documented in the description of the data set. Yeah. The atlases describe the healthy brain. Yeah.
It's possible that with a new niche or a new data, analyze with a probabilistic model or analyze a new data with this software or an algorithm. It's possible to analyze a new information or a new data with.
Of course, that's what you usually do. You combine it with new data, right? Or are you asking whether if you have new data, you can bring it into the system?
Yes.
That is also possible. That's a very good question, but also a tricky one. Maybe we could deal with it in the tutorial later on. So you could, in Zebra Python, in the Python client, when you run it on your computer, you could also bring in your own parcellation, for example, easily. Yeah. Of course, in the online viewer, it uses our prepared set. You cannot easily put in a parcellation there because that's an online system, right? But for example, in the online viewer, what you could do if you have a nifty image, and it is already in the MNI space, you could just drag and drop it on the browser window and it shows it is superimposed. That is not so much documented, but we will soon advertise this more because I repeatedly hear that this is useful. So you can actually combine your own data with that. When you use the Python version, it is actually very flexible, and there's also some documentation on it.
So now it is my pleasure to introduce you to Professor Nicola Palomero, who's also working in the Institute of Neuroscience and Medicine, in the Institute with Ferdinand and Timo. And she will tell us more about the more biochemical world moving around your brain and this receptor architecture, what is really going on in the chemical layer of your brain.
Right. So this is going to take a second.
We're
Just for So to do with the question about the brains, the brain, I mean, unless otherwise specified, if it's tissue from like for example, a biopsy from an epileptic patient, the tissue that we analyze is so it's healthy controls, meaning that it is body donors without a known history of neurological or psychiatric disorder, okay? This doesn't mean that the brains are necessarily 100% perfect, because as Timo said, they're from old people generally, because young people, I mean, the ideal thing would be if somebody very young died like that of a heart attack, you know, and then we would have a super short, and they did that, you know, in hospital, and then we would have a super short post-mortem time and everything would be perfect, okay, for research. Generally, young people die, let's say, maybe of motorcycle accidents, and then generally their head is a bit damaged, so we can't use their brains for, you know, research, right? So the brains that we can use for research generally come from older people, and older people, even if it's not you know, specified anywhere in their you know, by any doctor, because they didn't know. We often have small, what are called micro infarcts. So we might have, I might have, you might have, any of us could have just very small lesions in our brain that were caused by small bleedings that were just so small that they didn't cause any symptoms, okay? So that's why when we look at analyze our post-mortem brains that we use for mapping, in some of them, there are some little positions where we see, oh, here there was a little infarct at some stage during the life of this body donor, okay? But it was so small, as I said, that it wasn't big enough to cause any clinical symptoms, and that's then what we consider a healthy donor, okay? Right, so receptors. You've heard a lot about receptors in the EBRAINS atlas. Now, what I'm going to tell you, about over the next hour, more or less, is why is it interesting to analyze receptors, okay? And what do they do in the brain? What are they important for? Now, when we think about connectivity, very many people think about fiber tracts. They think about the axons of neurons that are connecting one brain region with the other brain region. And what they forget is that actually these fiber tracts, yes, they go from, you know, this part of the brain to this part of the brain. So these are all axons that are connecting these two parts of the brain. But axons are actually like dead-end streets, okay? There is no physical connection between the two neurons. So what we have is we've got one neuron, and then we've got its axon here, which would be running along the fiber tract, and information within a neuron is transferred via action potentials, which I will talk about a bit more later on. And this is what we talk about electrical conduction in the brain. But when we reach this point here,
the dead end of the street, right? We need, the brain needs to change the tactic it uses for transmission of information. And this point here is what is called a synapse, and it is a specialized part of the brain cells that enables them to communicate with each other via what is called chemical conduction. And what you see here on the right side of the slide is simply, you know, a zoom into this synapse, and we've got the so-called presynaptic neuron. That would be this one here, the one that is sending information, and we've got the so-called postsynaptic neuron, which is the one that will be receiving a message, okay? And the presynaptic neuron can transfer information to the postsynaptic neuron via release of so-called neurotransmitters, which are basically chemical molecules that will bind to a so-called receptor on the target, on the membrane of the target neuron, and binding of the neurotransmitter to this receptor will result in a series of changes, which would then mean that the information has been transferred, okay? And this space here between these two neurons, between these two membranes, is very, very small. It's only a couple of micrometers, if at all, right? It's called the synaptic cleft. But it's literally, you know, there is a space between neurons. So you've got to imagine, although when you look at a brain section, you'd think, well, this is something solid, that the cell nerves are actually not the cell nerves, sorry. The brain cells or nerve cells, they're not connected to each other. They're not touching each other the way two countries would be touching each other on a map of the mainland of Europe. We've actually got a whole load of very small, like islands, okay, that are very close to each other, but there still is water between each one of them, right? And that is what this synaptic cleft is. Now, if I zoom out again, what you see here is the postsynaptic neuron, the one that's going to receive the information. And here you'd have the axon of the presynaptic neuron. And in the brain, one neuron does not just receive input from one other neuron, as was the example in the previous slide. But what we have is a very dense interconnection, okay? So that brain cells can create like networks to enable brain function. And in this case here, the receptors that I drew, I've color-coded them because these different axons that come from different cells, these neurons, they don't all have to use the same neurotransmitter as the chemical substance that transfers information, right? We can have different kinds of neurotransmitters, which I've coded here in different colors. Now, in the brain, we've got different kinds of neurotransmitters, and there are the so-called classical neurotransmitters, which are the ones that I'm going to be talking about today mainly. And classical neurotransmitters their mechanism of action is such that the classical neurotransmitter it's synthesized in the cell body of the neuron. Then it is transported along the axon of the neuron till it reaches the synapse, and then it is released into the synaptic cleft, right? It's a one-way street. That is one of the characteristics or the main characteristic of classical neurotransmitters, okay? We've got non-classical ones where you also have release not via the axon, but via the dendrites. It becomes much more difficult to analyze these ones here functionally. So that's why today I'm going to concentrate on the classical ones. just the same as the glutamate receptor, the glutamate neurotransmitter can bind to different receptor types. Each one of the other neurotransmitters that I have listed here can also bind to different neurotransmitter types, and I've coded them here with different colors. And later on, I will explain to you why it's important that a given neurotransmitter can bind to different neurotransmitter types. So what happens when a neurotransmitter binds to its receptor? Basically, there is a change in the membrane potential, okay? Now, the membrane potential is basically a measurement of the difference in the concentration of ions that we find within the cell. That's the space that is called cytoplasm, right, inside the cell, and the extracellular space, that would be, well, you know, if you think of my analogy with the islands and the sea, the cytoplasm would be the island, and the extracellular space would be the sea, okay? Now, extracellular space and cytoplasm, they have different ions. Some of them have a positive valence, for example, sodium. Other ones have a negative valence, such as chlor or chloride. And in the cytoplasm, we also have proteins which can bind ions, and these are generally negatively valenced ions, okay? And the cell membrane is it has pores in it, so it's not completely let's say waterproof. It does allow for movement of ions between the extracellular and the space and the cytoplasm. Movement through these pores would be without energy consumption and would be a simple diffusion based on whether there are more ions of one valence outside the cell or inside the cell. Okay, so this movement is basically one where the concentrations try and become equal on both sides of the membrane. And then we can also have transfer of ions via certain molecules which would need energy. And in the cell, in the brain, when nothing is happening, when the neuron is what we call at rest, there is a balance between the ions that are outside and inside, such that we have a value of -70 millivolts, okay? And it's always so that the extracellular space has a higher concentration of positively valenced ions than the cytoplasm. And that's what results in this resting state potential of -70 millivolts, okay? And you can measure that. Experimentalists can measure that. And binding of a neurotransmitter to its receptor will simply change this resting state potential slightly. It will either make it less negative or even positive, or it can make it more negative, okay? So if the binding of a neurotransmitter to a receptor makes the membrane potential less negative, we talk about an excitatory postsynaptic potential, shortened as EPSP, and we talk about a so-called depolarization of the membrane, okay? And this is mediated by activation or by binding of a neurotransmitter to an excitatory receptor, okay? On the other side, we have so-called inhibitory postsynaptic potentials, IPSPs, and they are mediated by the activation of so-called inhibitory receptors. And the fact that the membrane becomes more negative is what we call a hyperpolarization, okay? Now, at a given synapse, we don't just have one receptor, we can have different receptor types, and the presynaptic neuron doesn't just release one neurotransmitter molecule, it releases very many neurotransmitter molecules, okay? So at any given time point, very many neurotransmitters are released into the extracellular space, and they can bind to the different receptors that are located at that synaptic position. So at one given time point, we can have a different number of EPSPs and or IPSPs, okay? Now, if the changes in membrane potential elicited by the EPSPs are the same as the ones that are elicited by the IPSPs, then they simply cancel each other out and nothing happens, okay? If, however, we have several EPSPs and at the same time, then their effects add up, and what we have is really a bigger increase in the change in the membrane potential. And if this increase is large enough to reach a so-called threshold value, which lies at around minus 50 millivolts, okay, then a so-called action potential is elicited, okay? And then we say that the neuron has fired and that information has been transferred from the presynaptic to the postsynaptic cell, okay? So activation of an excitatory receptor facilitates the production of an action potential, whereas activation of an inhibitory receptor, because it makes the membrane potential more negative, it brings it further away from this threshold value, it's actually making it more difficult for the targeted neuron to elicit an action potential, okay? Now, why do we talk about neuronal firing? This goes back to experiments in the 60s, which is basically, well, maybe for two reasons. Reason number one is that this building of an or eliciting of an action potential, it is a situation where once this threshold value is reached, there's no turning back. The neuron will elicit an action potential, okay? And the other reason is, well, the way that experimentalists made themselves aware of the fact that a neuron was firing. And what you're going to see here now is a recording that Hübel and Wiesel made of one of their experiments when they were trying to understand how cells in the primary visual cortex react to a stimulus, okay? So they discovered that single neurons in the primary visual cortex can be activated by a single visual stimulus. And they will only fire when this stimulus happens, okay? And this video here, I will get it to start, yeah. This video here, yeah. Good, oh. Okay, so you heard that like sound I was making. Okay, well, that was the firing of the neuron. And it was fired, so this neuron literally only fired when this white line was at this position here, okay? There are other neurons that only fire when a dot is presented and not when a line is presented, or they only fire when the line is presented in this horizontal direction or so on. Okay, so this firing, it almost sounded a bit like a machine gun kind of a sound. Okay, so different kinds of receptors means that of course you can classify them. We can classify receptors then based on their pharmacological characteristics, and then we would have, you know, glutamate receptors, GABA receptors, acetylcholine receptors. They can also be classified based on their outcome, and I already mentioned excitatory and inhibitory receptors. And they can also be classified not on what their outcome is, but how this outcome was reached, so by their mechanism of action. And here we have so-called metabotropic or ionotropic receptors. And the big difference between these two different kinds of receptors is that the ionotropic receptor is an ion channel. So if the neurotransmitter binds to the ionotropic receptor, the ion channel will open and ions will be able to flow through the membrane, okay? And ionotropic receptors, because they are ion channels themselves, they have a very fast mechanism of action, but it is very restricted. It is literally only where that one receptor is that you can make changes in the membrane potential occur. Metabotropic receptors act via a so-called second messenger mechanism. So the receptor itself is the protein that is embedded in the cellular membrane, and it recognizes the neurotransmitter. The neurotransmitter can bind to it, but then actually in the cytoplasm, we've got these second messenger systems, which can be different kinds of proteins, which I'm not going to go into today because that would be an entire lecture in itself. But these second messengers are the ones that ultimately result then in the opening of an ion channel and through which ions can flow, okay? They have a slower mechanism of action because this cascade of events has to happen before the ion channel opens, and it's also more widespread. And why is that? So what I'm showing you on here on the left, on the right, would be basically cross sections through the membrane, right? Where again, you know, up in blue we have the cytoplasm, and down here in orangey, up in blue we have the extracellular space, and in orangey we have the cytoplasm. And what I'm doing now in this view here in the center of the image is a view of the cell membrane that you would have if you were in the extracellular space, okay? And what you've got are here and here, you've got two ionotropic receptors, and here you've got one metabotropic receptor. And if the neurotransmitter binds to the ionotropic receptor, you've got the opening of one ion channel per neurotransmitter that is bound to a receptor, okay? But if here we have a neurotransmitter that binds to that receptor, we've got the opening of several ion channels, okay? So that's why it's slower, right? But it's also more widespread because several ion channels can be opened. And it also takes longer for these ion channels here to close again. So it's a longer lasting effect. So this means that if a given neurotransmitter can bind to different kinds of receptors, and these different types of receptors can have different effects or can mediate this effect via different mechanisms, it gives the cells a very high degree of plasticity by which they can transfer information. The transfer of information can be super fast, so you can, you know, run in somewhere and say, I need this now. Okay, I'm finished. I'm going now. You don't have to do any more. Or you can go in more slowly and you can say, I need it. And you can keep on, you know, saying I need it for a while, and then till you don't need it anymore, and then you go. And this just helps the brain modulate brain activity in very different ways, okay? Now, receptors. So they're not just super important, you know, because they enable brain function, but they also allow us to do mapping in the brain, okay? Receptors are either single proteins or protein complexes, so groups of several proteins. And there are different methods that you can use to visualize them. On the left of the slide, there is the so-called positron emission tomography, PET. It has the enormous advantage that you can visualize receptor distribution patterns in vivo, right, in a scanner. It has the big disadvantage that you have a relatively low spatial resolution, and also because of basically ethical reasons, there's a limit to how many receptor types per subject you're allowed to visualize, okay? Because this visualization requires injection of a radioactive substance into the person that you want to examine. So of course, if it's a patient and they need that for their diagnose, well, you know, you can do it, but for research purposes, you really have to, you know, explain very carefully why it's necessary and why this kind of information can't be acquired any other way. On the right side of the image, there's immunohistochemistry, which is basically the exact opposite scale regarding spatial resolution. So here you can look, if you do immunohistochemistry coupled with electron microscopy, you can look at single synapses. You can go down to the nanometer level as opposed to the couple of micrometers that you would see with PET. Disadvantage, it's post-mortem. And another disadvantage is that with immunohistochemistry, you can only visualize single proteins, okay? So if you want to analyze a receptor that's actually not built, not composed of a single protein, but that is a complex, well, then you can't visualize the complex itself, okay? And that's why in the middle we have the third method, which is the one that sort of like combines both possibilities. In vitro receptor autoradiography, which is the method that I basically use for my research. It has the disadvantage that it is a post-mortem method. It has quite an advantage that it has quite a high spatial resolution, and it has the super advantage that with this method you can visualize the receptors that at the time of death were expressed on the neuronal membrane, and at the time of death would have been able to modify the membrane potential, okay, because they can recognize the neurotransmitter. It also has the big advantage that it's fully quantifiable, which neither PET nor immunohistochemistry are. PET always requires a so-called reference region of the brain, and all receptor concentrations must be expressed relative to that reference value. Right, so what you see here are several sections through a human hemisphere. They're coronal sections, and they're through the central sulcus, so they're approximately through the central part of the brain. And each one of these sections has been processed for the visualization of one receptor type. The name of the receptor is written underneath the section itself, and then you've got you know, the neurotransmitter to which it binds. And color coding is such that blue colors code for low receptor densities, and the warm colors code for high receptor densities, okay? And anytime you see one of these images in the rest of the slides, this color convention will always be the same, okay? And even at this, low resolution, you can see that receptors are not homogeneously distributed throughout the brain. We have regional differences. We also have laminar differences. Okay, so there are parts of the brain, for example, where we've got very low densities compared to other parts that have higher densities. We've got these are subcortical structures, so caudate nucleus putamen, part of the basal ganglia. That here is the thalamus. Here we've got the hippocampus, got cortex. Sometimes cortical structures have lower densities than subcortical structures, and sometimes it's the opposite situation. Okay, now I'm going to zoom in a bit more. That's a drawing, showing you the section, a typical section of one of the ones that I had before. And now I'm just zooming in to the region around the central sulcus. This here is the central sulcus, and I'm only showing the M2 receptor. And this is what I mentioned before, that receptors, we can use them for mapping purposes. The central sulcus, we have one part here, which you can see has a very high density of M2 receptors, and the position at which these receptor densities change are highlighted by the arrows. And this high M2 receptor density is localized at a position that we know corresponds to the primary somatosensory cortex. And we know that because we have neighboring sections which we have not just processed for the visualization of receptors, but we've also processed for the visualization of a cell body staining, and that serves as our linkage point to other studies where we can see, obtain this information that we know that this is the primary somatosensory cortex. And here we've got the primary motor cortex. So M1 primary motor and S1 primary somatosensory. And if you compare the position of these three arrows here with the position of the arrows in the alpha-1 receptor, so alpha-1 are receptors for noradrenaline or for the serotonin 5-HT2 receptor, you will see that these arrows, which always indicate the position at which a significant change in receptor densities has happened, that they're always located so at approximately the same position. It's not necessarily it's exactly the same position, because although these sections look very similar, they're actually not exactly the same section. They're neighboring sections. They're only 20 micrometers thick, okay? And they're through the brain, so the human brain. So 20 micrometers in a human brain isn't that much of a distance, but it's different sections. So receptors, the differential distribution of receptors throughout the brain helps us find cortical borders. The important thing is that, as of here, I've shown you 3 examples where all 3 receptors show the border between these areas. But you must know that not all receptors necessarily show all possible borders in the brain, okay? And this is also important, as I will talk about later on, for brain function. I mentioned the cell body stained sections. We also have myelin stained sections, okay? And here again, you can see that the correspondence between changes in receptor density, the position where these changes happen, it is comparable to that of where classical cyto or myeloarchitectonic changes take place. And for example, you can see here that this stripe within the primary visual cortex, which is called the Genari stripe, this dark stripe caused by a stripe of very densely myelinated fibers running within the primary visual cortex and parallel to the brain surface, that is also highlighted here. You can see it in the GABA A receptor. It's the part where you've got the lower densities of the GABA A receptor, okay? This here shows another part of the sections that I presented earlier on. Now, instead of highlighting the central sulcus, I'm highlighting the region of the lateral fissure, and this little gyrus that you see here, this here would be the lateral fissure, and this gyrus that you can see within it is the so-called Heschel gyrus. It's the part of the brain where we have the primary auditory cortex, so the part of the brain where we first become aware of the fact, consciously aware of the fact that we have heard something. Again, here I've got the M2 receptor and the alpha-1 receptor. Here we've got arrows. We've got, oh, this arrow here, sorry. Well, I'll have to move him into its place. This arrow here should be over here. They are found at the same position, and here is where we've got an example that I talked about earlier on, saying that not all receptors have to show all possible borders. So at this position in the brain, for example, the NMDA receptor does not show this border, okay? But the NMDA receptor shows borders in other parts of the brain, okay? Now, if you're working in brain research, you want to do mapping because we want to know, how many parts, structurally distinct parts, is the brain composed of, right? But ultimately, we want to be able to link this structural segregation pattern with a function. And here is where receptors really become very helpful, okay? So they don't just show borders, but when you simultaneously analyze many different receptor types within, a given part of the brain, you can see that the differences in the densities of receptors in different brain areas tell you something about the hierarchical processing levels of those areas. And hierarchical processing levels in the sensory system or in the brain, it basically means what we have, what we call a primary area. That's the one where you first become aware of the fact that you have perceived a stimulus, right? Be it visual, auditory, or sensory. And then, but in the case of the visual system, just because you become aware of the fact that you've seen something, with that information, you can't say what you've seen. That information has to pass on from the primary visual system to the, from the primary visual cortex to the secondary visual cortex to other higher areas for you to be able to explain what you have seen. If you have seen a person or an object, if it is something that is moving or static, if it's in a color image, black and white, dark or bright, okay? And differences in receptor densities also distinguish between functional systems. So back here to our example of the central sulcus. On one wall of the central sulcus, we have the primary somatosensory cortex. And on the other wall of the central sulcus, we have the primary motor cortex. And if you compare the densities of different receptors on both walls of the central sulcus, you will see that we have always very different pictures. We have very high densities of some receptors on one side of the wall, and at the same time, very low densities of that receptor on the other wall, or, the opposite pattern, that we've got higher in M1 than in S1, okay? So we can distinguish between motor areas, or not motor areas, well, areas that are involved in motor functions, areas that are involved in the visual system. And this gets interesting when you move up from the so-called unimodal areas, the ones that are only involved in processing of one modality, up to the so-called association areas, where two or more modalities or more information comes in. And here's where then sometimes we know what modalities are important for that brain region, but we don't always know that, okay? And here's where receptors, analyzing receptors, would help us find that. And the third principle was this hierarchical processing levels that I was talking about earlier on. Here we find that many receptors have systematic changes when moving from primary through secondary, tertiary, up to higher processing levels. And the examples shown here are, again, on the left, the coronal section through the central sulcus, and here on the right, it's also a coronal section through a human brain, but through the occipital lobe, and we'd have the visual cortex. Now, most of these principles that I've talked about are evolutionarily conserved, as you can see in this slide here. So here we've got a section through the human brain. In the center of the image, we've got a section through macaque monkey brain, and on the right, we've got a section through a rat brain, okay? And in EBRAINS, you will also have receptor, so you will also find data on different species. And Timo mentioned that we have atlases for 2 monkey types, the macaque monkey and also the marmoset monkey, and there's also a rat atlas brain. And when we started looking at these very many different types of receptors, we realized that the important thing is to not just analyze a single receptor type, but to look at as many different receptor types as possible being as technically possible and also as financially possible, because this is unfortunately a pretty expensive technique. And we also realized that it would be important to visualize these receptor densities simultaneously, right? And looking through, you know, the different possibilities that I started looking at just bar charts, because all the papers that I'd read showed bar charts. But my problem was that I was looking, so when I was doing my PhD, I was looking at many different regions and many different receptors. And when I had bar charts, I had so many bars that I basically couldn't see anything anymore, right? So that's why I was working with Excel, and I just went into Excel to see what are the different possibilities of you know, depicting densities. And that's when I came upon this polar coordinate plot, where for every single receptor that you analyze, you've got one axis, where the scaling of the axis is the same, okay, for all of them. And then these dots basically indicate the receptor density of that given receptor, okay? And because for the human eye, if you want to compare, you know, different areas, looking at these clouds of points, it becomes a bit difficult. You can just join these points by lines, okay? So the lines don't mean that something is moving up or down. It's just joining these dots, okay? And because I'm very slightly neurotic and I wanted things to look nice and tidy, I got rid of the dots, and that's how the so-called receptor fingerprint was born. Yeah.
Which region is this from?
This here is A1.
Oh, okay.
Primary auditory cortex. But good question, because now I'm showing you the fingerprints of S1, the primary somatosensory cortex, and M1, the primary motor cortex. And here you see together, you know, as a plot, what I described before, the fact that I said that different functional systems differ in their neurochemical structure, okay? So the fingerprints of different brain regions differ in their shape and also in their size, okay? And we can use that for different kind of analysis. And these are numbers. I mean, right now it's just a polar coordinate plot, but this is created from numbers, and you can use those numbers for your statistics.
I also wanted to ask the y-axis, what are the values?
This is femtomole per milligram protein. Yeah, I did have a version of this slide with the labeling of the, yep, okay. Femtomole per milligram protein. Right, so these are differences at the regional level, okay? So the entire primary auditory somatosensory motor cortex If we go into a single area, I mentioned earlier on that we also have laminar differences in the distribution patterns of receptors, okay? And that is what this slide here shows. So it shows a little cutout through the primary visual cortex, and then at the top left, we've got the site architecture, the mile architecture. I've highlighted the Gennari stripe. And then what I've done is that I've transferred these two white lines here that you see in all the other snippets. They highlight the position of the Gennari stripe, okay? So you can see that although there is a relationship between the laminar changes in receptor densities and the laminar changes in the distribution of cell bodies or myelin fibers, there isn't necessarily a one-to-one correspondence. And that's because the cell body staining reveals cell bodies, but the receptors are normally localized on the dendrites, and the dendrites of cell bodies that are located here within cytoarchitectonic layer 4B, their dendrites can go up here into layer 3, or they can move down into layer 5. Okay, so that's why it's not surprising, but still we have a laminar correspondence. And we also have a canonical principles of organization within these layers. So here I'm showing you 4 different brain regions, and the cytoarchitectonic image and the M1 receptor, just as an example. And you can see that there are differences, you know, in the distribution of the M1 receptors in each one of these areas. But what we always find is that generally most receptor types have their highest densities in the superficial layers, the so-called supragranular layers, and lower densities in the deeper layers. Okay, that is one canonical principle of organization. And another important thing is that okay, so how do I say this? So this distribution of receptors is important because we also know that the superficial layers and the deep layers, they differ in their connectivity patterns, okay? So the superficial layers are the ones that are targeted by axons coming from the thalamus. They are also the layers that are targeted by axons coming from other cortical areas, and the deeper layers are the so-called output layers. And the axons, they go either to other cortical areas or they are cortico-subcortical projections, okay? So we have the higher receptor densities within the layers, the input layers of the brain, which makes sense because it's the one where we have to have the synapses to enable the information transfer, okay? Now, synaptic density, when we created this slide here for this figure, we only had one paper available for the human brain which reported distribution of synapses, and that was one that reported distribution of synapses in the primary visual cortex, and there they reported that the highest density of synapses in the primary visual cortex was located in layer 4 and deeper layer 3. And we thought, fantastic. And that's also the part where we find the highest receptor densities, right? So we wrote that there's a correlation, positive correlation between receptor densities and synaptic densities. In the meantime, I had the pleasure of being in a PhD defense committee of somebody who was looking at synapses in the human brain in the entorhinal cortex. Their data isn't published yet. And that rule that we found for the primary visual cortex doesn't apply to the entorhinal cortex. So you see, we still need a lot of research, okay? And hopefully, yeah, there'll be that article we published too. Animal models. I've basically been talking about the human brain all the time. I did mention that we've got macaque, monkeys, and rats. Animal models are necessary for research because there are certain things that we can't do in humans. And, you know, once you go through all of the ethical paperwork, or, you know, you are allowed to do research with animals, okay? And if you want to do research with an animal model, it's important that you know your animal model, okay? And that's why in my group, we have been working over the last years, you know, quite a lot on creating a map of the receptor distribution in the macaque monkey brain. So, you know, we've identified many different areas, created the fingerprints. This data is available in EBRAINS, and it's also visible in the EBRAINS atlas. And it's basically the work of my two postdocs, Meichi and Luthia. And we've also used this data because, you know, we don't just want to create a map, but we want to understand how this correlates with other structural aspects of brain organization and with function especially. We correlated these receptor densities with neuron densities, with dendritic anatomy. So, you know, if you have cells with a bigger dendritic tree than another cell, well, it's going to have more space for synapses, right? And we also compared it with track tracing data, so the axons going from area 1 to area 2 or whatever. And what we saw was when we analyzed all the receptor densities in all the areas that we identified, we extracted the fingerprints, we did statistical analysis. In this case, what I'm showing here is a principal component analysis. We could show that the PC1 segregated the areas along a so-called principal axis, where we had mainly areas involved in sensory processing on one side and association areas on the other side. And the main difference at the level of receptors and neurons and so on, the main difference of these areas along this PC1 axis was the fact that we had a higher number of receptors per neuron in these higher association areas than in the unimodal areas, okay? And when we look at the differences between the PC2, basically the big difference that we found was here for the 5-HT1A receptor. You had like a peak sticking out in some areas, whereas you had an indentation in other areas. And actually the difference between these areas here is the fact that these ones up here are, from the evolutionary point of view, older than the ones that are shown lower down in PC2, okay? So what we have is that PC1, our first principal component, is segregating sensory and association areas, whereas our PC2 is segregating evolutionarily older from newer areas, okay? They also help us segregate functional systems, right? So I've got all of these areas, these names, they're coded in different colors. And these colors are because these areas belong to different functional networks. Functional neuroscientists have often defined the 7 main functional systems. And here we've got the visual and the somatomotor one. And these are all areas that are located to the left here in my PCA analysis. And all of the other areas here, the dorsal attention, the salience, the default mode network, these are higher order functions. They are located to the right of the plot. So I don't just have a a segregation between sensory and association. I've got a segregation between like sensory input and then really cognitive functions, okay? And here, the areas that are segregated by the principal component 2, we've got areas that are more involved in working out what is going on in the world surrounding me versus what is going on inside of me, okay? So we've got this segregation of sensory, cognitive, and internal focus versus external focus, okay? These differences in receptor densities also correlate with, so basically the overall numbers of receptors. As I mentioned at the very beginning, we've got these more receptors per neuron type. So we have this linear increase in receptor densities. We also have a linear increase in the size of the dendritic tree when we move along this PC1, and we have a negative correlation with myelin content. So if we've got less myelin, we've got more receptors, and if we've got more myelin, we've got less receptors. And one thing that I found which was really nice was when we projected these PC1 values onto their location on the surface of the macaque monkey brain, we obtained this image here, okay? And when I saw this image here, it reminded me immediately of this other image here, which is it's a drawing of Brodmann's map, which I'm sure you've heard about earlier on. And Marcel Mesulam, in his pioneering work in year 2000, he postulated that in the brain you had different functional zones, and he gave those functional zones these different colors. And he said there's one functional zone that is the primary sensory motor one, which is colored in blue, and this is blue, and this corresponds to this part here. It also corresponds to this part here. You must know that in the macaque monkey brain, the primary visual cortex isn't just located on the medial surface of the hemisphere within the calcarine sulcus, which is the case in the human brain, but a large portion of the visual cortex in the macaque monkey brain is also on the free surface, on the lateral surface of the hemisphere, okay? And then we have the unimodal association areas, the heteromodal association areas, so these higher association areas, which are the ones that are here in orange. You've got here, you've got here, you've got this wonderful one here. This part of the brain we haven't analyzed yet, so that's why it's still empty. So this, it's sort of like reassuring for different reasons. One, it does make sense to use the macaque monkey, or to continue using it as a model for the human brain, and it is a good model for the human brain if you want to analyze what the meaning of receptors is, because not all animal models are good for all questions, right? Not all information can be directly transferred or translated to the human brain. And then the other thing is also that we had postulated from the beginning that the receptor fingerprint of an area tells us something about its function, because there's this link between structure and function. So the fact that this principal component analysis results so closely reflected the functional zones was something that we found very reassuring. You can use receptors, for example, also for modeling. This is something where I was only involved in as a co-author because I don't do modeling, but my colleagues in Montreal, they used the receptors from the human brain to inform their models and to estimate what different kinds of receptors could influence different parts of the brain, and perhaps also explain why that part of the brain is particularly vulnerable to, in this case here, it was Alzheimer's. And you can also use, for example, within a species, just for the healthy brain, you can use receptors for simulation processes. So, for example, here in this work, Sean could demonstrate that the different densities of dopamine 1 receptors that we find throughout the macaque cortex are the ones that enable the brain to concentrate on a single factor and not be distracted by a distractor, okay? And with that, I'd like to finish. I started a little bit late and I finished even later.
Thank you. That was really enlightening. I would like to know if you have considered using euthanasia patients from countries that allow.
We have.
Yes.
We have. I've got mixed feelings. So I have a study where we looked at receptor densities and gene expression levels in tissue from the same donor. And we got the data from the tissue from the Netherlands brain bank. And it's fantastic because it's the only one that has this super short postmortem time, which you need for the gene data. And it was, it's super valuable. I don't know if a person that wants to die can be considered normal. And I mean, no, normal, yes, healthy, in the sense, I think that, that life is something very valuable and you must be really desperate if you see no way out, if the only way out of whatever it is situation that you're in is death. So I'm not sure. I'm not sure. But yes, I've used it. I've used it.
I have a second question regarding receptor radiography, which is the tool you're using. It looks amazing. Is there any way to get a subcellular level where the receptors are in membrane?
Not with this method. There is currently no method that allows that. So something. So no, not at a large scale. Nothing. There is no. Well. At a large scale, you could look at single cells, very small tissue. You can use not that method exactly, but you can use a ligand that's bound to a gold ball and then do that, but you can't look at entire brain sections. So, yeah, there's no method that currently allows that at this scope.
Hi, thank you very much for your presentation. I'm sorry if it is a really basic question.
There are no stupid questions, just stupid answers. Go ahead.
I have no background on biology, so I have a question regarding electrical stimulation and what happens with the potential actions that you were explaining at the beginning. If you could explain me a little bit of what happens when you do the electrical stimulation with the potential and with the electrons and the things that you were explaining.
Do you mean electrical stimulation in the sense of well, you see electrical stimulation, there are loads of different kinds of electrical stimulation. Do you mean, for example, when you have implants,
yes,
deep brain stimulation?
Yes, and you stimulate in the area.
So what happens is the deep brain stimulation, what you're doing is you are, and how it mechanically works, I don't know exactly either. because I'm a biologist, I'm not an MD. But basically, with these deep brain stimulations, your electrode is it's
Adding the electrical activity.
Thank you. It's electrical activity. That was the word that I couldn't think of. And you're activating several neurons simultaneously, and you're basically from outside inducing a change in the membrane potential. So you are pretending to be a presynaptic neuron.
Okay.
And you then change the membrane potential of the several neurons that you are stimulating.
Okay.
And then there you induce the changes in the postsynaptic neuron, which would result in the release of neurotransmitter in other spaces.
Okay.
Would that be a good?
Yeah, perfect.
Well, I've learned something now, thank you. So I learned something from your basic question, thank you, seriously.
I have one question. When you were talking about the layers and your technique that you had, you could paint the layers with the different receptors, and you were talking that they are not homogeneous like the standard layer, because we are talking about the receptors that are normally located on the axons, not on the soma of the neuron. So it would make a different map if you could bring it just like the distance. This has been tried to locate the soma of the, to try to calculate after this result that you have where the soma locations will be according to the physiological characteristic of the neurons that have those receptors?
Well, listen, if you're doing electrophysiology, you have a completely different situation, because what you have there is you have your brain tissue in culture, and you stick your electrode into your neuron, and then you can, in your culture medium, you can add a certain concentration of a neurotransmitter, right? And then you see what happens to the membrane potential of the cell that you're measuring. If you're looking at, but then you don't know, well, right, so you know that I must have a receptor for that neurotransmitter, but do I have one receptor or two or 20 in that thing, right? So you can't quantify it. The question you are asking is something that, with one of the grant proposals that I'm just submitting this week, that we want to try and answer. What we want to try and do is say, okay, we know that in this part of the brain, in layer 5, we have this kind of neuron, this kind of neuron, and this kind of neuron. And we know from other studies that this kind of neuron has a very long, thin, dendritic tree that reaches all the way up to layer 2. And it has a layer 5 or a layer 4 neuron that only reaches into the bottom part of layer 3 or whatever. So we want to integrate information from different kinds of findings and say, what do we know about the morphology of the cells that we are analyzing, and then it will be a sort of a probability map. So up here,
Yes.
we've got a, we've probably got more dendrites for this kind of cell than for that kind of cell. So it's located with a higher probability on that kind of a cell than on that kind of a cell. Yeah. But yeah, so right now, but good question, grant proposal.
Hope you get it.
Thank you, me too. Thank you.
Two more quick questions, and then because we have to move with the plan. So
When you showed the laminar structures of the receptor densities, it looks sometimes that it's a bit oscillating on the pattern.
Yeah.
Is this, I don't know, measurement inaccuracy, or could you also estimate, I don't know, there's a cortical column and therefore we have a certain band or so.
Oh, yes. Well, yes, yes. What you see here are actually, this is the primary visual cortex. The primary visual cortex we know is not homogeneous. It's organized into blobs and interblobs. And what you can see here are these differences in the blobs and interblobs.
Brain atlases starting on the post-quantum brains. Because I'm not familiar with, so I'm by training, I'm computer scientist and I'm in computational neuroscience. So I'm interested to know when all the sensory inputs, visual inputs, the auditory inputs, auditory inputs are off, what's the level of precision we are dealing with these atlases and localization of regions?
When they're off.
So then,
oh, so how do I know that a certain region is the primary visual region? And
I mean, when there is no visual input to the person's brain, when it's dead.
Yes, but so obviously when the person is dead, there's nothing, there's no cognitive function, there's nothing going on, right? But that part of the brain was the one that processed the visual input when the person was alive. And God, how do we know that? Well, we know that from animal studies, where we can put electrodes into the brain. We know that from studies with humans. So, for example, epilepsy patients that have to undergo a really tight screening before the operation to know exactly what part of the brain is the one where the focus of the seizures. So yeah, I mean, when the brain is, when you're dead, there's no function. But that part of the brain was responsible for a given function when you were alive. It's a post-mortem, so I can't see any function-related changes. I can't see any changes related, for example, with the use of a drug. You would be able to see that if you did a PET study, an in vivo study, but you wouldn't get this spatial resolution, and you would definitely not be able to, you'd only be able to look at maximum two structure or molecules in one subject. There was one more question. Is there a lunch break now?
Yes, but I just wanted to mention, maybe it's also the very nice thing of having the atlas, that all the information that we have collected through time about where the function of the specific regions is located also very functional in brain. So it's collected into one thing. So when we put the brain in the same space, the same map, we can say, okay, actually that region should be approximately the area where this function was located. So this is also one of the great things about the atlas, that it allows us to find ourselves also when we cannot test this function, right?
Exactly. Or you can do it a different way. I mean, maybe you've got a part of the brain from a functional imaging study, and you want to know what does the receptor architecture of that brain look at. So then you can go into the atlas and overlap them. Is it a short question?
Very short question.
Did you?
We talk
Afterwards. Okay.
It's more is related to the vitro models, or maybe your opinion about you know, or develop the work in the in vivo or in animal model. But my question is about the in vitro. For example, the NAM platform, the organoids, and it is possible to model the specific regions or sections of the brain, and to analyze the neurotransmitters and the protein in a specific model using, for example, the cells of the donor patients, and the person using pluripotent cells. If this idea to create platform would be translated to the neuroscience research?
I definitely yes, but I think that is like still at the beginning. So you know, when people started doing modeling, they just at the beginning they model like a single protein. Then people went on to model 2 cells. And whoa, I've got a model with 2 cells. Then people started modeling because you know, modeling computation advanced. They were modeling an entire cortical column. And then they said, well, and I've got a cortical column, and if I make 10 cortical columns, I've got an entire brain. And then they thought, well, no, it's not such a good idea because the brain isn't just made of 10 identical cortical columns. Maybe I should make my cortical columns a bit different. And this is the way then modeling has been advancing. So definitely, I think it's the way to go. I think we need modeling because there are questions that we want to answer that we can't answer with a human brain. We can't answer with experiments in animal models or in animal, you know, models of humans, and we will need computer models for that. I am not sure how developed, for example, organoid modeling is yet, but it's on a good way, so to speak. I think it's worth trying, you know.
Okay, so thank you very much.